Hello, welcome to Cell Biology Online. Hope you're doing well and excited about taking cell biology in your pajamas. Don't even have to leave the bed. But first, let's go over some announcements. Lectures are going to be available Tuesday and Thursday mornings, and I will have a link that will send you to YouTube so that it doesn't take as much of your computer hard drive and space, things like that. In terms of what chapters we'll be covering, I posted a revised syllabus that you can take a look at on Canvas, and that will have the dates and the different chapters we'll be going over. Most of the lectures, or most of the chapters, will still have pre-class quizzes, and I'm just keeping their normal due dates. In-class activities, you'll be able to submit those online. You can type them up, or you can print them off and write on them, take a picture, send it on Canvas, whatever you like. If you want to work virtually with a partner, that is okay with me as well. And I will probably do some type of survey for every assignment where you can select some questions that you want to go over in more detail, and then I can talk about those before the assignment is actually due. So I still want to have a chance for you all to go over problems that are difficult or maybe that I haven't worded the best, possibly, and so that you guys can still be getting pretty good scores on those. Exams, we have 2 remaining exams. They're CHI-TESTer exams. They're the same as usual in terms of average size and relative number of multiple choice questions to essay. So I don't expect to change them too much. You will have a limited time to take them. So what I would recommend is to find a quiet space where someone's not going to interrupt you. Go through the easy questions first and then go back to the hard questions. I don't care if you want to go online or check on your book or your notes to answer those, but again, remember you don't have unlimited time. So the way I'll calculate that is I've given these exams before on CHI-TESTer, and I have an average amount of time that it's taken students, and I will probably do that and then add some extra time on top of that so that you don't have to feel like you're rushed. But if you wanted to look up the answer to every single problem, you wouldn't have enough time to do that. Labs I've revised, so we will no longer be doing cell culture. I have some virtual labs that we're going to be doing, and those will have a post-lab quiz to make sure that you did the activity and that you understood it. There will be a separate video that I will put out on what your lab report is going to be on, how to analyze the data from the cell culture experiment, a rubric for you to go over, and all the different timelines for that. But again, you don't need to worry. I've extended deadlines for everything so that you'll have enough time to write everything. We'll still do lit labs, but you won't have a group presentation. Instead, everybody will be assigned a figure or a part of a figure, and you'll fill out the worksheet that you normally do. Then I will go through those, make any edits or corrections, and post all of your worksheets on Canvas so that you can all look at them. And then a few days later, I'll have some type of assignment for you to do to kind of check your understanding of the material. And so that's what we'll do instead of a presentation. There will be no more lab notebook checks, which I'm sure we're all excited about. You can keep your lab notebook. I would still use it to study. And of course, I will adjust any questions on the exam that covered labs we were going to do and take those out and replace them with ones about the labs we are going to do. If you have any questions whatsoever, I am accessible by email. I could also talk on the phone. If you would like to talk to me on the phone, just email me and I can give you a phone number to call. Otherwise, again, just like normal, ask me anything, any questions that you have. So today we're going over chapter 12. We've learned a little bit about membrane structure, and now we're going to talk about how the membrane acts as this semi-permeable barrier. How does it, once it decides to let something through the membrane, how does it do that? and what are the implications of letting things through the membrane or not. There are 3 objectives for this chapter. The first is to understand how ions and small molecules can be transported through cell membranes, and then to be familiar with the 2 main types of transport proteins and how they work. The largest portion, or I think the newest material for you, will be our third objective, understanding how nerve cells communicate. So we're going to be learning about voltage differences across the membrane, how that can lead to membrane potential and action potentials, how neurons can send signals to each other via these action potentials. So on the bottom of this page, there's a link to a website that's a great study aid. I've posted this PowerPoint slides of this lecture online, so even if you can't click on the link in this YouTube video, you can still go and get the link yourself and find it. There's a variety of different activities, little mini games you can do on there, and I would recommend that you do the Build a Membrane and the Energy and Transport Challenge. So for our first objective, we're going to talk about how small molecules and ions can travel through the cell membrane. What is permeable? So we know already that small nonpolar molecules can travel on their own through the cellular membrane. They can just diffuse right through. However, if a molecule is polar, it is going to go much more slowly through the plasma membrane. If it is large, even if it's unpolar, if it's really large, it's going to take time. If you have a polar molecule that's large, that's going to be even worse. And then lastly, ions. These are positively or negatively charged molecules. They are going to not travel through the cell membrane very well at all. And so you can see some examples. Gases like oxygen, carbon dioxide can travel through the cell membrane very easily. Water, ethanol, and glycerol are some small uncharged polar molecules that can slowly travel through the plasma membrane. And then we have things like amino acids, glucose, the sugar that our cells need, nucleosides. Those all are going to be pretty well repelled by the plasma membrane. And then lastly, those ions that we talked about. So when they show, for example, here with ions, they've got this arrow kind of bouncing off. That is saying that it can't travel through the membrane, right? Well, in fact, things like ions, they can travel through, but it's very, very slow. So in order to get something like an ion across the membrane, it's better to just use a transporter. So what I would suggest that you do is pause the video for a minute, go through these problems on your own, and decide, will it diffuse and why? So you're thinking about size and polarity for these. All right, let's start with this indole molecule. Is this polar or nonpolar? And do you think it's large or small? Right? So this is a relatively small molecule that is nonpolar. We've got those hydrocarbon rings, right? So this will be able to diffuse through the membrane. Next we have ATP. You can see that this is larger than that indole. It also has, depending on the pH of the solution, you can have a negative charge on some of these phosphate groups. And so for this one, we're going to say, no, it will not diffuse because it is large and polar. Potassium, this is an ion, and it has a positive charge. So despite the fact that it's really small, it will not travel through the membrane because of that charge. Next, we have fructose. This is a type of sugar. Fructose is going to not travel through the plasma membrane because it is too large. Then we have methane. Methane is a gas. Most gases can travel through the cell membrane. And this is just like those others. It is nonpolar. Next we have octane. Octane, this long hydrocarbon. You can guess that that's going to be nonpolar, but it is a lot bigger than methane. And so this, it will somewhat travel through the plasma membrane. It'll be pretty slow though because it's so big. Next we have glycine. This is a relatively small molecule, and so that will allow it to travel through the plasma membrane. Ethanol, we've got not too giant of a molecule. It's relatively nonpolar on one side, but we do have this oxygen on the other, which is going to give it some polarity, and so it again will somewhat be able to travel through the plasma membrane. In other words, it can, but it's going to be slow. Lastly, we have carbon dioxide. This is a symmetrical molecule, so you can guess that it is probably not polar, it's nonpolar, and it is pretty small. And so yes, this will travel through the plasma membrane because it is nonpolar. So let's talk more about ions. So ions, depending on the type, can be very concentrated inside the cell or outside the cell. And so you will need to know for these ions listed here, these are common ions in mammalian cells, whether they are found at a higher concentration inside the cell or outside the cell. So first we have sodium. Sodium is highest outside of the cell. The concentration is lower inside of the cell. Potassium is the opposite. So potassium has a higher concentration inside of the cell than outside. Calcium has a high concentration on the outside of the cell, as well as chloride. However, hydrogen has a higher concentration inside of the cell. Now, for the cell to avoid being torn apart by electrical forces, these opposite charges must be balanced. And so when I say electrical forces, you can imagine, right, we know that opposites opposites attract and like charges repel, right? And so we need to make sure that these are balanced so that you don't have, say, this really strong attraction across the membrane that could rip the cell apart. So we know that outside of the cell there's a lot of sodium, right? And so there needs to be something that's going to balance the sodium. So sodium is going to have a high positive charge outside the membrane, which means that there would be more of a negative charge inside of the cell. And so chloride is the ion that tends to help balance out sodium. It's got a negative charge, and we see that chloride is also outside the cell. And so it's kind of neutralizing that positive charge outside of the cell. So unfortunately, I think my face is in the way of this slide, this part of the slide in the corner, and I don't really know how to move it. I will work on trying to figure that out for the next video. So the high concentration of potassium that is inside the cell, that's going to give a large positive charge inside the cell. So that can be balanced out by other macromolecules in the cell, such as DNA and proteins. A lot of these macromolecules have a negative charge, and there's a lot of them more inside the cell than outside. And so that helps balance that positive charge inside the cell. So here I've drawn a representation of a beautiful cell, and we can try and imagine the relative amounts of these different ions inside and outside of the cell. So again, first of all, we have sodium that has a higher concentration on the outside of the cell. So you can imagine that sodium, if it's going to travel down its concentration gradient, would want to enter the cell. Potassium has a higher concentration inside the cell. So according to its concentration gradient, it would tend to want to leave the cell. Next, we have calcium. Calcium is kept at very low levels inside the cell relative to outside the cell. Calcium is a really important signaling molecule, so it's very important that it's kept regulated and very low. Then we have chloride. Chloride is found at a higher concentration outside of the cell. And then lastly, hydrogen is found at a higher concentration on the inside of the cell. So again, as a reminder, what is a concentration gradient? This is a chemical driving force, and it is caused by differences in the concentration of a certain type of molecule across a membrane. So in A, we have more of this molecule inside the cell, so it wants to diffuse out, if possible, to that lower concentrated area. And in B, we have the molecules are at a higher concentration outside of the cell, and so they have this driving force to go into this area where the molecules are more diffuse. Again, molecules want to go from an area of high concentration to low. And as we've talked about before with mitochondria, when you have molecules that are trying to go from a high area to a low, and there's a membrane in the way, this generates potential energy. Let's talk about membrane potential. So before we're talking about chemical differences, concentration gradients, now we're talking about differences in charge, because each of these ions also has a charge, positive or negative. And so it can cause an imbalance in the charge across the membrane, and this is called a voltage difference. On the left, we have an example of a membrane of a cell, for example. On the left, we have in blue is the inside of the cell, and on the right, outside of the cell. And we can see that regardless of what ions they are, there's pretty equal balance of both positive and negative ions on the inside and on the outside. So there's no potential here because the positive and negative charges are equal on both sides. However, on the right example, we can see that inside the cell there are more cations, more ions that have a positive charge than negative. And so the inside of the cell has a slightly positive charge. And on the outside of the cell, we can see that there's more ions with a negative charge. And so the outside of the cell has a slightly more negative charge. So a more positive charge on the inside, negative on the outside. That's a voltage difference, and we say there is a membrane potential. The resting potential is the normal voltage that a cell has when it's unstimulated. Interestingly, the membrane, when a cell is at rest, is not zero. In fact, it's actually between -20 to -200 millivolts in animal cells. In other words, that means it's slightly more negative inside the cell. And we'll talk later about what that means, but you should know that the cell's resting potential is always slightly negative. Let's talk next about transport proteins. There are two main classes of transport proteins. First of all, we have transporters, and second, we have channels. So transporters are going to be very selective in what they allow to travel through. And the molecule that's going to be transported has to fit into a very specific binding site. And that's what allows it to be selective. Channels are less specific, but they do have the ability to discriminate based on size and charge. So what determines whether a solute moves in or out of a cell? You can remember we've talked about the concentration gradient, right? So if something is traveling down its concentration gradient, that is called passive transport. Second, we talked about these voltage differences, right? The relative amount of positive and negative ions, cations and ions on either side of the plasma membrane. So if, for example, the charge is slightly more negative inside of the cell, that means that solutes that have a positive charge in the cell's surroundings will be attracted into the cell. All right, so what if you wanted to move a molecule against its gradient? So against its concentration gradient. That is going to require energy, and so we call that active transport. Here we have some examples of three different types, or a variety of different types of transport. So first we have simple diffusion. Something like a small nonpolar molecule can simply travel down its concentration gradient, and in this case, for example, go into the cell. Next we have channel-mediated transport that we talked about, and transporter-mediated movement. So if the molecule is going through that channel or that transporter down its concentration gradient, it doesn't require energy, and so that's called passive transport. The molecule is still either too large or polar to get through the plasma membrane on its own, so it has to have some type of facilitation, but it doesn't require energy because of that concentration gradient. Now, if you are trying to move a molecule against its concentration gradient, so for example, we want to move these blue blocks outside of the cell where they are found in a higher concentration, you're moving that molecule against its concentration gradient. It's going to cost energy, and so that is called active transport. Again, the net driving force across the membrane is composed of the concentration gradient and the membrane potential. This is called the electrochemical gradient, which is a pretty cool word that combines both of those things, the concentration and the membrane potential, because we have to be considering both of those things. So here we have an example of an electrochemical gradient. In A, we have inside of the cell, we've got more of a negative charge than outside of the cell. And so these ions, or cations, these molecules that are outside of the cell with a positive charge, will flow down their concentration gradient, but they will also be attracted to the inside of the cell because it is more negative inside the cell. So here the concentration and the voltage gradients are working together. Next, we have an example in which they're working in opposite directions. So we have more of this molecule inside the cell than outside. So due to the concentration gradient, you would say it wants to leave the cell. However, in this case, the outside of the cell has a positive charge, and like charges repel. And so even though that molecule wants to leave the cell, it's being repelled by that positive charge on the outside. And so not as many of those molecules are going to actually cross the membrane. Next, let's talk about aquaporins. These are a unique type of channel that allow water to cross the cell membrane. So you've probably heard of osmosis before. This is the idea that water tends to move from an area where there's low solute concentration to an area of high solute concentration. So if you think about a cell in a tissue, where do you think that the solute concentration is highest? Inside of the cell or outside of the cell? The solute concentration is highest inside of the cell because you can think of all these different things that are packed inside of the cell. We've got all these macromolecules, right? All the organelles. And so because of that, water will tend to want to flow into the cell. And so it will flow. It can travel through the plasma membrane, as we talked about at the beginning, but relatively slowly. If that cell has aquaporins, these channels that allow water to travel through, the water is going to travel through the aquaporin into the cell. Now, if the cell doesn't do anything about that, it will swell and swell and swell and eventually explode. And so cells have a variety of coping mechanisms so that they don't do that, ways that they can kind of expel water or balance the concentration of solutes. And we'll talk about some of those in the future. So every cell membrane has its own set of transporters. There's transporters on the surface of the cell, and also there's organelles inside the cell that have transporters. So take some time to go over these problems and quiz yourself. Where would you expect sugar transporters to be located? ADP transporters and hydrogen transporters. Think about where the cell is getting sugar from. Typically that is coming from the bloodstream, so outside of the cell. So you would expect to see a transporter for sugar molecules to be at the cell surface. ADP, diphosphate, that has the opportunity to get a third phosphate group to become ATP. We know that the majority of ATP is made where? In the mitochondria. And so they're going to be transporters for ADP at both the inner and the outer mitochondrial membranes. Lastly, for hydrogen, we know that the pH is very low in lysosomes, and one way that they're able to keep that pH really low, well, pH has to do with concentration of hydrogen, right? And so there are going to be hydrogen transporters on lysosome membranes. Let's look at an example of a transporter in the cell, such as the glucose transporter. So glucose is a sugar, right? A carbohydrate. It's important for providing energy to the cell. So food, after it's ingested, it's broken down. So you had a sandwich, right? You've got carbs in that sandwich, broken down into glucose molecules, and eventually gets to the gut. And then those glucose molecules are transported via villi on the inside of the gut. Then from those cells along the intestine that make up the villi, they're going to transport that glucose to the bloodstream. And then the glucose in the blood can go to all of the cells in the body and be transported into those cells that need it. So there's no electrical component, no membrane potential that's involved in glucose transport. It is just a concentration gradient. Here we have an example of a glucose transporter at a cell membrane. And we have a lot of glucose on the outside of the cell, not as much on the inside. So the concentration gradient is going in. And so in this transporter, we have glucose. Again, transporters are selective, so the glucose has to be fit in the specific binding spot, and then it can be released into the cell. And in this example, this would be passive transport. Here we have some examples of active transport. We can have something such as, not necessarily active transport, but transport against concentration gradient. So one of those could be our active transport, such as the ATP-driven pump that we have shown in the middle. On the left, we have a coupled pump, and this is pretty neat. So we're trying to take this orange molecule, and transport it across the cell membrane against its gradient, right? Well, if you have another molecule that can travel down its gradient via that same pump, that same transporter, this transporter can harvest the potential energy of that other molecule flowing down its gradient to pump a partner molecule against its gradient. And we'll talk about this coupled pump in more detail. Lastly, this is kind of a neat one. We have a light-driven pump. There's a molecule in this transporter that responds to light, and when light hits it, it changes conformation slightly. That shifts the structure of that transporter and helps to transport that molecule against its gradient. So you can probably guess what part of the body would have these light-driven pumps. It's going to be cells in the eye that receive light. Let's talk about active transporter, the sodium-potassium pump. This is a super important pump in mammalian cells, and so you should know how this pump works. This is an ATP-driven pump. It's going to be the pump that keeps sodium at a low concentration inside of the cell. So we said that most cells has sodium at a low concentration inside. So the sodium potassium pump is constantly pumping sodium outside of the cell against its concentration gradient. This is also a potassium pump. So while sodium is getting pumped out, potassium is getting pumped into the cell. And both of these are movement of molecules against their concentration gradient or electrochemical gradient. Here we have a cartoon of this pump. You can see it's using active transport, so it's using ATP to power the movement of these molecules. We have 3 sodium ions that are going to be expelled out of the cell and 2 potassium ions that are going to be transported into the cell. I would encourage you to take a minute, either now or afterwards, and watch this video about the sodium potassium pump, because again, it is very important for you to understand how it works. Let's talk more about coupled pumps. So again, this is the idea that you have potential energy generated by a concentration gradient or electrochemical gradient of one molecule that can be harnessed to help transport another different molecule across its concentration gradient. So you can have symport or antiport. Symport, both of the molecules are going in the same direction. Antiport, they're going in opposite directions. This video that I encourage you to watch describes how sugar can be transported across the membrane in a situation where it's going against its concentration gradient. Now this is a method of transporting sugar. This method is not using ATP. So you should be able to understand why it is that glucose or sugar can be transported against its gradient and how that can happen without ATP. Sodium is often the molecule powering coupled transport. So if you watched the video on the previous slide, they talked about how sodium can power the transport of glucose against its concentration gradient. And this is only possible because of that important sodium-potassium pump. It keeps a low concentration of sodium inside the cell and a high concentration outside the cell. That way, other pumps that need help can use sodium as that helping molecule to help out to pump a molecule across the membrane. So again, many transporters of other molecules are indirectly coupled to this sodium-potassium pump. That means that if something goes wrong with the sodium-potassium pump, many other pumps will stop working and the concentrations of other molecules will be impacted. Now this sodium-potassium pump we said is an active transporter. It uses ATP. In fact, it accounts for 30% of the cell's energy consumption, which is pretty impressive. So in the gut, you can imagine that there may be situations where glucose has to travel against its concentration gradient. Maybe you had a snack and that glucose made its way to the intestine, and these intestinal cells took up that glucose. So now they have a lot of glucose in the cell. Then maybe you have a cookie in a little bit, and now you still have more glucose that has to get into those cells when there's already a lot. Right? So how can you transport glucose across its plasma, across its concentration gradient, right? So this is again kind of showing another representation of that video. So on the luminal surface, the side that lines the cavity of the gut where the food is, we have these glucose transporters. So they are coupled with sodium transport to power the movement of glucose against its gradient. So in this example, we have the inside of the cell has a lot of glucose. The outside of the cell has a lot of sodium. So that means that sodium wants to travel into the cell. So in order to force glucose to go against its concentration gradient, we can have co-transport of sodium down its concentration gradient, right? So they are traveling together across the plasma membrane. This would be an example of symport. Here we have another representation of one of those gut epithelial cells. You can see the little villi on top, and there is in green on the top of the cell, we can see our sodium-driven glucose symport. That's what we just talked about. Now the glucose eventually does have to leave those epithelial cells in order to get into the inside, to leave the cell to go to, say, the bloodstream. And so in that case, the cell is able to use passive transport. If there's a lot of glucose in that intestinal epithelial cell, it's free to leave when it wants down its concentration gradient through that passive glucose uniport. Again, in order for this glucose to enter into this cell, it is dependent upon a gradient of sodium. And so we have here shown our important sodium-potassium pump. Right? So on the right, again, just summarizing what's happening to this glucose. The cell is able to maintain a low concentration of sodium due to that sodium-potassium pump. And because of that, the sodium is always wanting to enter the cell. And so the potential energy from that sodium can be used to power transport of glucose into the cell. Let's do another problem. So in muscle cells, calcium is an important signal for them to contract. So as we said before, calcium levels are kept very low inside the cell at all times. And this is important in muscle cells as well. Whenever the calcium rises in these muscle cells, they are going to contract. So there is an ATP-driven calcium pump that pumps calcium back out of the cell. And however, in the heart, they are going to have an additional calcium transporter to really help make sure that that calcium gets out really quickly so that the heart can pump very fast. And so in this case, we have an antiport that exchanges calcium for sodium. All right, so we have a calcium-sodium transporter. The majority of the calcium ions that enter the cell during contraction have to get rapidly transported out, and this antiporter helps the cell to do that so that the muscle can relax. Now you may have heard of the drugs Ouabain or Digitalis. These are used, Ouabain and Digitalis. These are used for treating people that have heart issues, and it makes the heart muscle contract more strongly. Now the way they do this is they inhibit the sodium-potassium pump. Right? So we've got an ATP-driven calcium pump, we've got a calcium-sodium antiporter, and we have the sodium-potassium pump involved. So how do you think that this drug works? And what would happen if you get too much of this drug? So take a minute, pause the video, think about what you think the answer is to this question. All right, so this one is a little bit complicated, but I know you guys can get this figured out. So what's going to happen? You inhibit the sodium-potassium pump. That means that you're not going to have this nice concentration gradient of sodium. Normally, sodium is low inside the cell and high outside the cell. And so if you don't have a pump that's maintaining this gradient, eventually the levels are going to equalize relatively on the inside and the outside. Now, what's the impact of that if you don't have this concentration gradient? Well, we know that the concentration gradient is important for calcium to get pumped out of the cell. We have this calcium-sodium antiporter, right? And so now that calcium cannot harness the potential energy of sodium trying to come back into the cell. to pump calcium out, to be pumped out of the cell. That means that calcium is going to remain at higher levels for a longer period of time inside these cells. And when it receives its next signal to contract, there's already going to be elevated calcium. So it's already going to be up high. It's going to get even more calcium, which is going to lead to an even higher concentration of calcium inside that cell. So it's just going to build up even more calcium, and that's going to result in a stronger and longer lasting muscle contraction. So that's good, right? Again, but not necessarily if you take too much, right? So if you take too much, what's going to happen? Well, we know that that sodium-potassium pump is important for a variety of things, not just this calcium transporter. For example, we talked about glucose transport across the membrane. If you start screwing across all the cells of the body with their sodium-potassium transporters, you're going to have a variety of other molecules get imbalanced in cells across the body, and you're going to have problems. So I wanted to go over this problem with a diagram because normally I would write it out on the board, and that's not possible. So again, we have my beautiful cell here, and we have normally we've got a low concentration of sodium inside the cell and a high concentration outside the cell, and the same thing for the calcium, right? So we have a sodium calcium transporter, and I don't know where the calcium went, but it was right here. This pump is going to pump calcium out of the cell. So contraction has happened. The cell needs to remove calcium. So calcium is going to be pumped out of the cell, and that is against its concentration gradient, right? Because there's a lot of calcium outside the cell. And so that means that it has to have something coupled to it, something to power it. And so we've got sodium that is at a high concentration outside of the cell relative to inside. And so it's going to couple movement of sodium down its concentration gradient into the cell with movement of calcium out of the cell against its gradient. Now this is only possible because sodium is being constantly kept at a low concentration inside of the cell. And that is done by, over here on the right, our important sodium-potassium pump. And this is an active transporter, so it uses ATP. It pumps potassium into the cell and sodium out of the cell. And this Wabane or digitalis drug is impacting that transporter. So what's going to happen? You're going to have an increase of sodium inside the cell because it's not getting pumped out by that sodium-potassium pump. Now you don't have as much of a concentration gradient across the cell membrane, and you're going to have less potential energy for pumping calcium out of the cell. Right, so there's our calcium-sodium transporter being inhibited, and that's going to lead to a rise in calcium inside the cell. Right, we're going to have already higher levels to begin with. Even more calcium is going to get added the next time the heart gets stimulated, and so the heart's going to contract harder and longer. Let's talk a little bit about channels. So channels, they are going to allow the passive flow of molecules across the membrane. So here we have an example on the right of a channel, right? So channels don't do active transport. They just open, and whatever can flow through that molecule is, whatever molecule can flow through that channel is going to flow down its concentration gradient. So here we have an example of a potassium ion. Now channels are still relatively selective. You can see that it gets quite narrow in the center. So a molecule that's not the right size will not fit. Also, you can't see it in this cartoon, but that protein, that channel in green, it's got all these side chains sticking out of it. And those side chains can interact with whatever ion is trying to travel through the channel and say, okay, are we going to let you through or not? Right? Most channels, when you think of channels in the cell, are ion channels. Right? So again, the diameter and shape of that ion channel and those amino acids that line the channel are going to determine whether or not the correct ion is present and whether it can flow through or not. Now there are some gated ion channels, right? So ion channels are not always just open all the time, right? So they can be like a gate that sometimes is open and sometimes is closed, depending on the stimulus, whether or not the cell gets the right secret password, right? So here we've got in this cartoon an example of an ion channel that has a gate. So membrane potential, we talked about this before, that is due to voltage differences, right? Differences in charge across the membrane. And we just said that channels are the main type of proteins that transport ions across the membrane. So that means that channels are important for determining membrane potential. And electrical signaling, when we talk about neurons sending signals, this is due to changes in this membrane potential. Now, earlier we said that the membrane of an animal cell has a resting potential of -20 to 200 millivolts. It's not zero, and we're going to talk about why that is now. So normally, the negative charges of organic molecules in the cell, so our proteins and DNA, things like that, are balanced by potassium in the cell. There's a high level of potassium inside the cell. However, the plasma membrane has these potassium leak channels. So leak channels are critical to understanding why the resting potential in an animal cell is slightly negative. These leak channels randomly open and close, right? So when it is open, what's going to happen? Potassium is going to rush out of the cell. If potassium, this positively charged ion, is leaving the cell, what does that mean about the charge inside the cell? It's going to become more negative. And that is why the inside of the cell is slightly more negative at rest than on the outside, and why we say it's been measured to be -20 to -200 millivolts. Here we have a cartoon that demonstrates this. So on the left, we have inside the cell, we've got leak channel, or sorry, we've got leak channel is closed at the plasma membrane, all right? And on the right side, we have when that leak channel is open. Right? So normally on the left, inside the cell, we have a high concentration of potassium inside in the cytosol, shown by those yellow gold diamonds. And when the channel opens, it's randomly flickering open. When that potassium leak channel opens, the potassium is going to rush down its concentration gradient and it's going to leave the cell, right? Now, it's not going to all leave the cell. Eventually, the charge outside the cell will become slightly more positive. And we said that like charges repel, right? And so eventually it's going to be balanced out. That concentration gradient and the voltage gradient are going to balance out. So even though it's not zero, it's close to zero. It's slightly negative. This is a very important concept for you to understand. There's only 3 objectives to this chapter, and like I said before, the neuronal signaling membrane potential type stuff is probably pretty new. And so I would encourage you to watch this video that goes over how resting membrane potential works. So when the membrane potential is negative, what exactly does that mean? Is the extracellular space or the cytosol more negatively charged? Hopefully by watching that video, you can understand that that means that the cytosol, the inside of the cell, is more negatively charged. We talked about ion channels being gated and that they have to receive a certain secret password or stimulus. And here's some different types. I think these are really neat. We've got voltage gated, ligand gated, and mechanically gated ion channels. So voltage gated, if the membrane potential changes, and it will sense that change and that will cause the the channel to open or close. Ligand-gated channels will only open if a specific molecule attaches to it. And then lastly, we have mechanically gated. These have to be physically moved in order for the gate to open, for that channel to open. Here's an example of that mechanically gated ion channel. I don't expect you to memorize this. I just think it's really cool. So looking inside the ear, we have these auditory hair cells in green, and they are interacting with the tectorial membrane in yellow via these stereocilia. And these stereocilia contain channels that are mechanically gated ion channels. And these stereocilia are all connected via these linking filaments that are attached to these channels. And so the tectorial membrane is going to vibrate when there is a sound wave. That vibration is going to hit those stereocilia and make them move. And when they move, that pulls on that linking filament, pulls on that channel, and causes enough force for that channel to open. That's going to allow ions to rush into the cell, and that will cause enough of an electrical charge, a change in membrane potential, to give that cell a signal to send to the brain. And that is how we can hear. So mostly what we're going to talk about, though, are voltage-gated ion channels and ligand-gated ion channels. So what I said before, voltage-gated channels, they are going to respond to changes in membrane potential. In this example, we have when the inside of the cell is negative, the ion channel is closed. But if the membrane potential changes and the inside of the cell becomes more positive, then the ion channel will open. Right? So it's like these channels have a voltage sensor. Now, what happens when one voltage-gated ion channel opens? Well, it's transporting ions. Ions have a charge. So that means that it can further change the membrane potential of the cell. This can have an impact on other voltage-gated ion channels nearby in that same cell. Right? So this spread of ion channels, all opening or all closing, is what we consider the basis of this electrical charge or electrical signal that travels down this length of a neuron. There's also yes, we've got an ion channel, changes the membrane potential. Eventually that change in membrane potential reaches another ion channel further down the cell. That ion channel opens, changes its membrane potential locally, and it's just this chain effect. Right? So let's talk about how this voltage difference of the membrane can help nerve cells to communicate in a little more detail. This link to this video, you could watch it before, but I would also watch it after because there's a lot of information in this video and it really nicely sums up what this third objective is going to talk about. So, neurons, my favorite cell type, of course. So their job is to send signals, they receive signals, they interpret and integrate a variety of signals. And so you have peripheral nerve cells. Those are the ones that are going to be receiving input and also going to be sending input. So feeling that you touch something hot, and also the signal that tells your muscles in your finger to move away. And then you also have your central nervous system, and so that's going to be the spinal cord and the brain, and they are what are going to be transmitting those signals. So you touch something hot, the peripheral nerves send a signal to your spinal cord or your brain or spinal cord interprets that and decides you need to move your hand. And so it sends a signal down the peripheral nerves to tell that muscle to contract to move your hand away. So the general structure of a neuron, it's not a blob like most cells that we've been looking at. It has a very long projection called an axon, and these can actually be quite long. So there are nerves that run from your spinal cord all the way down to the tip of your hand, right? Very long. Um, so we've got axon. Where those axons end, we have these branches, nerve terminals. And then we have on the other side of the cell body where the nucleus is found, and we have dendrites. And these dendrites are what can pick up signals from other neurons. So you can see with the red, the arrows are indicating signals are being received by the dendrites. The signal is traveling down the axon and being released at the nerve terminal. So signals along these axons are sent via action potentials. These action potentials have to be really fast, right? Right, we said there's a neuron, right, that can be the length of your arm. And so these ion channels, again, are what are important for this membrane potential change to travel down the length of a neuron. Now, you can have a local change, right? So here we've got an ion channel. It sensed a change in the membrane potential and it opened, and it let some ions through, and that changed the charge of the membrane. But that's only locally, just kind of close by that ion channel. So the further away, the less of an impact that single ion channel is going to have. So there is a signaling mechanism that the cell uses to help maintain that change and to amplify it so that it goes down the length of the cell. So a local electrical stimulus in one spot can trigger an explosion of electrical activity that travels down the entire cell. And when we say that a membrane potential or a change in potential is traveling down the cell's length, we call that propagating. So you may have seen that word before, maybe not, but propagation is a common word that people talk about in neuroscience and cell signaling, and it just means to reproduce or spread. So this signal happens, the change happens locally at one ion channel, and that local change is getting sent to another ion channel further down the cell membrane, and then to another one even further. And so it's continually renewing, the signal is getting continually renewed all the way down the length of the cell. So this signal is called an action potential. So whenever you have a change in membrane potential that's big enough to actually affect the entire plasma membrane of a cell, that is called an action potential. So again, we said that changes in membrane potential are regulated by ion channels. Action potentials specifically are regulated by cation channels. Okay. So we said that in a cell, normally the resting membrane potential is slightly negative, right? So if you have a neuron that gets stimulated, that membrane potential becomes slightly less negative, or in other words, a little bit more positive. When the inside of the cell becomes a little bit less negative, that is called depolarization. So polarizing, like opposites, right? So in the cell, when it's really negative, we say it's polarized. When it becomes slightly less negative or it becomes more positive, that's called a depolarization. So that's an important term to know. Here we have a graph in which the membrane potential is indicated on the y-axis. You can see we can have a positive or a negative membrane potential, and in this cell, the resting membrane potential is slightly negative at negative 60 millivolts. And then on the y-axis, we have time. So with a stimulus, some type of signal, that's going to depolarize the cell, depolarize the plasma membrane. It's going to become slightly more positive. Okay. So if this depolarization is large enough, if that charge on the inside of the cell increases enough, this will cause some sodium channels to open. So these sodium channels are voltage gated, and they're waiting to see, is the membrane going to become positive enough? And if it does, they will open. Now, what will happen if a voltage gated sodium channel opens? We know that sodium is normally really low inside the cell, so it will rush out. Or sorry, in. So sodium normally is really low inside the cell, so when a voltage-gated ion channel opens, the sodium is going to flow inside into the cell down its concentration gradient. So we've got a bunch of positive ions coming into the cell. That's going to make it slightly less negative or more positive, which will depolarize the membrane. Right? So that will make the membrane potential, right, even less negative. That means that if there's a voltage-gated sodium channel nearby, it's going to sense that depolarization. It's going to say, oh, I should open too. So it opens, and what happens? More sodium rushes in, and even further depolarization. So this just continues, right? It self-amplifies. And so we see that indicated on our graph, that the membrane potential just shoots up, right? And now we have an action potential forming. This happens really quick. Within a millisecond, the membrane potential has changed by about 100 millivolts. We can see it was around negative 40 and it jumped up to positive 40. So that's actually 80. No, if we start at minus 60, then it is, yeah, I did about 100. Obviously, I haven't learned how to edit videos yet, so hopefully they will get better with time. All right, so action potentials, they are going to be mediated, again, we said, by these voltage-gated cation channels. So we need to eventually turn off this signal, right? So these sodium channels have an automatic inactivating mechanism. It's almost like they have a little timer in them to tell them when to close. Right? So after a certain amount of time, the channel will change shape so that it is inactive. Right? So this is about a millisecond. So that means that even though the membrane is still depolarized, that channel is going to close. And it will not reopen until the membrane potential gets back to that resting state of a slightly negative charge. Here we have on the left an example here. So we've got our voltage-gated ion channel in green, and it's closed, and the membrane potential is slightly negative at rest, and it receives a signal, we can see on the right, that depolarizes the membrane. Now the inside of the cell is slightly positive, and so it will open up, right? It's going to let that sodium rush into the cell, but then within a millisecond, it's going to inactivate. You can see a little part of the protein changes shape, and so it will not reopen again until the membrane repolarizes or goes back to that resting value, resting level. Okay, so that is what helps to contribute to the downward slope of this graph to return the membrane potential back to its resting state. Let there be light. It's getting a little dark. All right, so here we have another demonstration of this voltage-gated sodium channel changing conformation. So on the top, we're just showing when the cell was stimulated, right, at 0 seconds. And within half of a millisecond, we can see that the membrane potential has dramatically increased, right? And that is because, you can see on the bottom, that sodium channel has opened. Sodium has rushed into the cell, made the inside of the cell slightly more positive. And then within a millisecond, it's going to inactivate. It's going to kind of get inactivated, right? We can see in the light green. And then eventually, once the membrane potential goes back to normal, it will close. And now it is ready to open again. In green on this graph, that slope, we've got a flat green line. That is what would happen if we had no depolarization that was sufficient to open these sodium gated channels, these sodium voltage gated channels. Now, unfortunately, just closing a sodium channel is not enough to bring the levels back to normal, right? So normally we have low levels of sodium inside the cell compared to outside. We just had a bunch of sodium rush in. Simply closing those channels isn't going to get those sodium ions back out, right? So what else could help with this? Take a minute to think about this one. Well, hopefully, this is a little tricky one. Hopefully you thought about something like a pump, right? So, or some type of channel, right? We need some way for that membrane potential to return to normal, right? We've got a more positive charge inside the cell, right? And we want it to become more negative. So what we have are voltage-gated potassium channels, right? So the potassium channels are going to open in response to depolarization as well. But potassium is different than sodium, right? Where is potassium, more potassium found? That would be outside the cell. Sorry, inside the cell, right? So these are going to open, right? When the membrane gets depolarized, there's a lot of potassium inside the cell, and so it is going to rush out, all right? So we've got potassium flowing out of the cell. We're losing this positive charge. Now the membrane potential is going to become more negative. Here we have a cartoon of our potassium channel, right? Normally there's a lot inside the cell. This is a voltage-gated potassium channel, so when the cell is depolarized, that's going to cause it to open, and the potassium is going to rush out of the cell. That's going to help to repolarize the membrane, and it was slightly positive, and we're going to bring it back more negative. All right, so that is what helps that action potential, that peak, to then go back down with time. And then eventually the sodium-potassium pumps that are in those cells will help to get rid of that extra sodium. So what would it look like if we had no voltage-gated ion channels amplifying the initial depolarizing stimulus? So first of all, which ions are doing this? It would be sodium, right? So we've got sodium voltage-gated channels. If they weren't for some reason responding to that stimulus, we wouldn't be able to get an action potential, right? And that's what we have this shown in green. We wouldn't have this propagation of the signal down the cell. Let's learn a little more about how this signal gets sent along the length of the axon. So we have from left to right is just kind of over the length of an axon. And on the top we have at time of 0, and on the bottom we have 1 millisecond later, right? So you can see that just a small part of the cell has been depolarized. It received a signal, the sodium, voltage-gated sodium channels opened, sodium rushed into the cell, made the inside of the cell slightly more positive, and that gave it a positive membrane potential. And what you see is that that is going to act on the ion channels to the right, right? They're going to sense that change and they are going to open. Now, 1 millisecond later, you can see that those channels that opened are inactivated, right? And that is going to help to depolarize the cell on the side of the cell where it received a signal, and then on the other side, it's going to propagate that signal down the length of the axon. So again, this is also a bit of a difficult concept to understand. And so I have a link for you, a video for you to watch that explains this step by step. And I highly encourage you that you watch that. All right, so we now have an understanding of how a signal gets sent from one side of a cell to the other. But what about from one cell to the next, right? How are these action potentials transferred from one neuron to the other? Where these neurons interact are called synapses, right? Now, neurons are not physically touching, they're just very, very close. And so there's a gap called the synaptic cleft in between those two neurons. So on the left, we've got a neuron that's received a signal at the dendrites. That signal spread as an action potential down the length of the axon and then terminated at the dendrites of another cell. Right? And so you can see that synapse there. There's a gap when you look close up where those two neurons interact. These neurotransmitters are going to induce a signal that will cause an action potential to travel down the length of the second neuron, which we call the postsynaptic cell. And eventually it will send a signal at its synaptic terminals to another cell. So what we have is an electrical signal, then being converted into a chemical signal, which is these neurotransmitters, and then it's being converted back into an electrical signal again. So these neurotransmitters are molecules that get secreted by the cell by exocytosis. Again, neurotransmitters, they are going to be found waiting at the synapse, the presynaptic cell, just waiting to be released. Right? So we have in yellow some nerves that are synapsing onto another neuron in blue. And so we can see these little circles are the vesicles where these neurotransmitters are stored, these little packages. And when they receive the signal, they are going to be released into that cleft. And then we can even see in electron micrographs, right? Those little gray tiny circles inside of the cell are the synaptic vesicles. One of my favorite neuroscientists is Santiago Ramon y Cajal, and he won the Nobel Prize way back in 1906 with Camille Golgi. Yes, that's what the Golgi apparatus is named after. There's also a Golgi stain. And he was the one that really proved or actually really proposed, was a proponent of the idea that these neurons are not one big web and that they're not physically touching, but they are just really, really close. And he called these protoplasmic kisses. He was very poetic. Now, this is a drawing that he did on the right here. Just really pretty work showing how these different neurons are interacting at synapses. And he actually, even though he won the Nobel Prize with Camille Golgi, they very strongly debated whether or not these neurons were continuous, all touching, or contiguous, very close together but not touching. And in fact, they both gave their Nobel Prize speeches arguing against each other over this topic. So again, at the synapse, we have the electrical signal, the action potential, being converted into a chemical signal, which is made up of neurotransmitters. These neurotransmitters are found in synaptic vesicles that fuse with the plasma membrane and release their contents into that gap or synaptic cleft. Now, these neurotransmitters are only able to be released due to voltage-gated calcium channels. Right? So the neurotransmitters are waiting in these vesicles, and when these calcium channels open in the neuron, that then allows the neurotransmitters to be released. So let's look at that in more detail. Here we have on the top in yellow our presynaptic nerve. So that nerve, it just sent us action potential down the axon, and it's getting to the synapse, the nerve terminal, and it's going to convert that electrical signal into a chemical signal that will cross that cleft and reach the postsynaptic cell, the cell that it's targeting. So we have our neurotransmitter enclosed in a vesicle, waiting at the end of this axon, and we have a voltage-gated calcium channel that is hanging out and just closed when the cell's resting. Now, when the action potential, this depolarization reaches the end of the cell, that is going to open our voltage-gated calcium channel, right? So it responds to a depolarization, right? And that opening of the calcium channel is going to indirectly lead to this neurotransmitter being released. So, which way will the calcium travel? Calcium is always kept low inside the cell, which means that the calcium is going to rush in when the calcium channel opens. So increased levels of calcium are what tells the synaptic vesicle to fuse with the membrane and to release its contents into the synaptic cleft. Now we have this chemical signal that is going to be received by the postsynaptic cell, and it's going to convert that chemical signal back into an electrical signal, right? So how do you transmit between chemical and electrical? Well, we talked about the different types of gated ion channels. In addition to voltage-gated ion channels, we also have ligand-gated ion channels. So we can see that the neurotransmitter has traveled into the gap, that cleft, and that is binding to an ion channel, a ligand-gated ion channel. So the ligand in this case is the neurotransmitter. And that is going to open that channel, and ions are going to rush in, probably sodium, right? That is going to depolarize the membrane because a positive ion is coming into the cell that's making the membrane potential slightly more positive, right? That's going to cause a change in membrane potential, and that can cause an action potential to initiate in that postsynaptic neuron. Electrical, chemical, back into electrical again. Take a look at the link at the bottom of this video to learn more or to review this concept. And I gave away the answer already. Yeah. All right, so these transmitter or ligand-gated ion channels in the postsynaptic membrane, right, the cell that's receiving the signal, are going to convert that chemical signal back into an electrical signal again, right? It's very important to remember. We have an electrical signal, the action potential. Then we have a chemical signal that allows these two cells to communicate, and that chemical is the chemical is made up of neurotransmitters. And then the next cell, it is converted into an electrical signal again. All right. So this is why neurons can send signals so quickly, right? That change in membrane potential can happen so fast. Right? So here we've talked about this already, right? Let's talk about these neurotransmitters in a little bit more detail. So neurotransmitters can be excitatory or inhibitory, right? So in the earlier I said, what if that ion that traveled into the postsynaptic cell was sodium? That would depolarize the cell. That would be excitatory. It's making that target cell more likely to have an action potential, right? But sometimes you may have a neurotransmitter that can be inhibitory, right? So inhibitory neurotransmitters bind channels, and that makes the membrane potential more negative, right? You don't need to understand the mechanism of how it works, but there are neurotransmitters that are inhibitory. When these ligands bind to the ligand-gated ion channels, instead of causing an action potential or depolarizing the membrane, it makes the inside of the membrane more negative. It's going to be less likely to respond and have an action potential. For example, we can have a chloride channel, right? A ligand-gated chloride channel. So chloride is normally kept low inside the cell. So if it opens up, if a chloride channel opens up, that means that the inside of the cell is going to become even more negative because chloride, the negative ions, are rushing in. Here's some examples of excitatory and inhibitory neurotransmitters, and you may have heard of some of these before. And I don't expect you to memorize those. Let's do a problem, right? So Curare causes muscle paralysis by blocking acetylcholine receptors at the neuromuscular junction. This drug was used by South American Indians to make poison arrows, and surgeons use it to relax muscles during an operation. Right? So we've got an acetylcholine receptor and a neuromuscular junction. So a neuromuscular junction is simply where a neuron is sending signals instead of to another neuron, it's interacting with a muscle and it's telling that muscle to contract. We talked about those as being peripheral nerves. So Curare is blocking acetylcholine receptors that are on the surface of these muscle cells. So take a minute, try to figure out how this could work, and then come back. Here we have the acetylcholine receptor. All right, this is on the surface of the muscle cell. It is a ligand-gated ion channel. So when acetylcholine binds to this ion channel, it opens, and it is specifically a sodium ion channel. Okay. And so we're going to have sodium rushing into the cell when it is open. Right? So that's normally what happens. Acetylcholine, we said that was an excitatory neurotransmitter. When sodium rushes into the cell, that's going to make the inside of the cell slightly more positive. That will depolarize the cell and promote telling that muscle cell to contract. Now this curer, right, what it does is it goes in and it takes the place of where acetylcholine would normally bind. So it's blocking this ligand-gated ion channel from interacting with acetylcholine. So what that means that no matter how many times that neuron fires, no matter how much acetylcholine it releases into the synaptic cleft, that ion channel is never going to open, sodium's never going to rush into that muscle cell, and it's never going to depolarize. Right? And so that is how you can cause muscle paralysis. Let's look at another example of modifying a ligand-gated ion channel's response. So a lot of psychoactive drugs are going to be working on receptors for neurotransmitters. So they're going to be impacting how these ligand-gated ion channels work. Now we don't know exactly how Prozac works, but we do know that it is an SSRI. Right? Selective serotonin reuptake inhibitor. So we know that it works, but exactly why this is beneficial, we don't know. So we have a nerve terminal. It's released serotonin into the synaptic cleft. Serotonin binds to a ligand-gated ion channel on the postsynaptic cell, and that causes an action potential on that target cell, that postsynaptic cell. Now, the longer the neurotransmitter is hanging out in the cleft, or the more neurotransmitter there is, in theory, the more signal you can promote in that postsynaptic cell, right? The more ion channels it can bind to. So here on the right, with Prozac, we have more serotonin hanging out in the synaptic cleft that is going to cause more likelihood, a greater likelihood or a greater frequency of action potentials, sending signals down that postsynaptic neuron. The way it works is there are other channels or transporters that will take up serotonin or other neurotransmitters and reuptake them back into that presynaptic cell. And that just helps to regulate the amount of neurotransmitters in the cleft because you don't want them hanging out forever, right? So Prozac, when they say it's a selective serotonin reuptake inhibitor, means it's preventing reuptake of neurotransmitter in the cleft. So there's more serotonin hanging out in the synaptic cleft and more excitation of that postsynaptic cell. I always forget that I write these out for you guys. All right. Now, there is not just one neuron interacting with another neuron, one single neuron. In fact, a single neuron can receive tons of signals from other cells. In this picture, we have a cell, and in red are all of the synapses, right? So you can see there's a ton of synapses on this cell, which is also cartooned in white. And so it's kind of interesting because what that cell now has to do is it has to process all of those signals. So it can be getting excitatory signals from one synapse, another one could be sending an inhibitory signal, and so it is able to combine, interpret, and record all of these signals. All right, so action potentials, those are really quick, right? Synaptic signaling, neurotransmitters, that slows things down a little bit. However, if you didn't have these synapses with these neurotransmitters and all these different combinations of signals to receive, the neuron would not be able to integrate all of these signals. In addition to integrating signals that it's receiving, neurons can adapt based on past use. Right? So you may have heard the phrase, use it or lose it, right? This is talking about synapses, right? So a synapse can adjust the magnitude of its response based on how often it's gotten signals in the past. And so this is called synaptic plasticity. It's also thought to be kind of the underlying mechanism behind certain addictions. So a synapse can adjust how it responds for a short period of time or a really long period of time. And so this is how you're able to learn, because your synapse starts to adjust. If you're doing, say, the same movement over and over again, that synapse can adjust and kind of strengthen that connection. All right, the last thing I want to talk about is this really neat thing called channel rhodopsin. So we briefly mentioned light-gated ion channels, right? And these are really great. They've been used in a variety of different research fields. And one of the biggest things that they've been really helpful for is to understand like circuits and how neurons are connected and why. So we know that, we've got all these neurons in the brain and there's specific circuits, but we don't have all of them mapped yet. We know that neurons are receiving input from a variety of different cells. They have to integrate those signals, what those cells are that they're having to, what cells are sending messages to them, and then where those cells are signaling to. All of this can be figured out, or has been figured out, using this tool called optogenetics. And so what optogenetics allows you to do is to control neurons with light. So scientists found this channel rhodopsin in green algae, photosynthetic green algae. So part of the photosynthesis process relies on light, and these channel rhodopsin are what receive that light signal in the green algae. So when light hits this channel rhodopsin, it's going to open, and it's an ion channel, so that means ions are going to flow in or out of the cell. So if you want to regulate whether a neuron is activated or not, you need to regulate its membrane potential. And so if you can regulate an ion channel in that neuron, then you can regulate its signaling. And so scientists figured out the gene for channel rhodopsin, and they stuck it into neurons. So they could put channel rhodopsin into a certain class of neurons, light them up, and see when that specific class of neurons get turned on, what behavior do they see in the animal. So they've actually been able to control the activity of animals, which is kind of freaky. So in this mouse, they introduced the gene for channel rhodopsin into a subset of neurons in the hypothalamus. So they wanted to know, what do these neurons in the hypothalamus control? What behavior? So these neurons in the hypothalamus will get activated if they shine this light on the brain. So what scientists do is they actually make a little window in the skull of the mouse, and so it's with a little bit of glass, and so they can attach a fiber optic cable to the head of that mouse and have a long cable that goes out to some power source, so the mouse can walk around just fine, and you can shine a light on its brain whenever you want. So in this case, the channel rhodopsin specifically responds to blue light, and when they shine blue light on cells, any cells that have the channel rhodopsin exposed on their surface are going to depolarize. So in this case, it is the hypothalamus, neurons in the hypothalamus. When the blue light hits the channel rhodopsin, it opens, it allows sodium to enter into the cell, causes a depolarization, which leads to an action potential, right, activating specific circuits, specific sets of neurons in the brain. So what they found was that these neurons were important for regulating aggression. The scientists put a glove, they just kind of blew up a glove and put it inside of the mouse cage, and the mouse is just like, okay, something new, interesting, whatever. But then when they turned on the light and activated those neurons in the hypothalamus, that mouse went berserk and started attacking the glove. But as soon as they turned the light off, the mouse stopped attacking the glove and just didn't care about it anymore. So yeah, pretty creepy, but also really neat, a really good way for scientists to understand the jobs of different neurons in our brain. All right, so that is all for this chapter. I will send out another video on Thursday at some point talking about the lab report and what to do in terms of that. And we'll also go ahead and take a look. You can go ahead and take a look at the virtual lab. Again, please email me if you have any problems. And if you want to chat by phone, just let me know and I can give you my phone number. Have a great day.